We are here to share with you what we've learned and just how far we've come since starting our master's in translational research. Reflecting back on the past 2 years, neither of us had any idea that we would end up where we are today. And that being said, we are so excited to present our capstone project, Translational Research Through a Commercial Lens, Improving Diagnostic Methods for Urinary Tract Infection. We first want to mention that although we are a two-person capstone group, this project does have a third team member, Jaspreet Randhawa. She has dedicated so much of her valuable time and effort to this project, and we are so grateful to have her as part of our team. In this presentation, we'll begin with a background and an overview of the capstone process, then discuss the two key components of this project, which are business development and the clinical study, and then end off with some future directions. So, what is translation? Translation is the process of turning knowledge, observations, and discoveries into interventions that improve human health. The aim is to produce more meaningful results and move innovation more quickly and efficiently to the people that need it. In other words, translational research is where research comes to life. The process of translational research is often classified by different stages of translation, which range from basic science research, as you can see on the left, to implementation and dissemination research, as you can see on the right. Our capstone project falls here, in what is commonly referred to as bench to bedside research. And now I'll pass this over to Jess to provide some background information on our project.
All right, so let's get right into it. A urinary tract infection, also known as UTI, occurs when bacteria colonize the urinary tract and cause an inflammatory response, often eliciting symptoms such as a burning sensation while urinating. There are fairly common infections that are treated with antibiotics. If you have a UTI and are not pregnant or have no fever, you have what is called a simple UTI. Complicated UTIs are UTIs that fall outside of this criteria. If you have a UTI and are an immunocompromised person, it is classified as a complicated UTI. Complicated UTIs also include UTIs that are associated with instrumentations such as catheters and also urinary obstruction such as kidney stones. In these instances, there's a much higher incidence of mortality, recurrent infection, and more severe complications such as sepsis. Several populations that are disproportionately affected include kidney surgery patients, older adults, and people with spinal cord injury. We first learned about complicated UTIs when we attended a seminar on spinal cord injury given by Praxis Spinal Cord Institute in December of 2021. James Hektner, who's also with us today, who lives with spinal cord injury, shared some of the unique challenges that people with spinal cord injury face regarding complicated UTIs. We were initially shocked to learn that UTIs are the second most common cause of death for this population. Then we learned that this is in part because impaired sensation can make it difficult to recognize the onset of a UTI, which causes delays in seeking medical attention, leading to advanced infection and severe UTI complications. Similarly, people with spinal cord injury, who often rely on catheters to void, experience UTIs more frequently and are more likely to develop antibiotic resistance compared to the general population. The combination of these risk factors can make UTIs extremely dangerous. After this initial conversation with James, we spoke with clinicians to identify challenges faced by healthcare providers who care for patients susceptible to complicated UTIs. They confirmed that UTIs are especially challenging to diagnose in patient populations that have difficulty communicating or do not exhibit or perceive UTI symptoms in typical ways. They also confirmed that UTI tests often give false positive UTI indications. In fact, several studies have shown that urine dipsticks are not reliable indicators of infection, giving false positive UTI indications almost half of the time. At this point, we investigated why these tests perform so poorly. We learned that conventional UTI tests are dipsticks that detect the presence of leukocyte esterase and nitrate in urine. These biomarkers elevate not only in the presence of UTI, but they are also elevated in the presence of asymptomatic bacteriuria, or ABU. There are two key things to know about ABU. First, ABU is harmless bacteria in the urine unaccompanied by any symptoms of UTI. And second, that treating ABU is not recommended, except for exceptions that were on the slide. ABU poses a unique challenge for populations vulnerable to complicated UTIs, and as these populations have a high prevalence of ABU. ABU is seen in over 50% of older adults over the age of 80 and 90% of people with spinal cord injury. Likewise, many people vulnerable to complicated UTIs do not necessarily exhibit typical signs and symptoms of having a UTI. For example, 29% of older adults experience delirium when they have a UTI. Without symptoms to rely on, it becomes even more critical for UTI tests to have a high level of diagnostic accuracy. One meta-analysis identified that 40% of ABU cases are misdiagnosed as UTI and treated with antibiotics. Alarmingly, antibiotic exposure is the biggest risk factor for antibiotic resistance, which kills over 1 million people every year. Considering that needless treatment of ABU accounts for over 17% of all unnecessary antibiotic prescriptions, we became aware of the significance of addressing this issue, not only for people prone to complicated UTIs, but for the general population on a global scale. After many months of consulting with stakeholders and reviewing literature, it became apparent to us that to address this pressing issue, there is a need for a more reliable UTI diagnostic method that can distinguish between UTI and ABU. Our ultimate goal is to develop and deliver a highly accurate diagnostic tool that addresses the limitations of existing UTI tests. We recognize that commercialization is a fitting approach to achieve this goal. Of course, this vision will take many years to execute, and for this reason, the objective of this year's Capstone project was to build the foundation for a startup dedicated to the clinical development of this innovative diagnostic method. Building this foundation involved 4 major components. First, acquiring funding to support research and development efforts. Second, learning how to protect intellectual property. And third, gathering information on prospective end users, the market, competitors, and regulatory pathways. And lastly, conducting a proof of principle clinical study to validate the use of alternative biomarkers for UTI diagnosis. Now I'll pass it back over to Siobhan to talk about the first 3 of these 4 components.
So when we started this project, we created a budget that included each of the components we intended to accomplish during the first year of operating our startup. Then in June of 2022, our team was awarded $25,000 for placing first in the Praxis Ideation Challenge hosted by the Ideation Clinic. And thanks to this initial funding, our project became financially viable. At this point, even though we did have enough money to cover all of the costs we would incur during the course of this Capstone project, we knew that running out of money is one of the main reasons why startups fail, and that we would need to be proactive and persistent about obtaining funding in order to achieve our long-term objective. We continued to identify sources of funding and ultimately have managed to secure over $40,000 to date. Another area that we invested a lot of time into was intellectual property. Considering intellectual property is often considered the most valuable asset of a startup, it was important for us to stop and gain a strong understanding of how to protect intellectual property before we generated any. We had numerous conversations with the University of Toronto's Innovations and Partnerships Office, as well as our mentors from the Forge and Health Innovation Hub, to learn about institutional IP policies, patenting, and commercial contracts such as NDAs and service agreements. We were advised to file a provisional patent in order to secure a priority filing date. And based on this advice, we conducted patentability searches to examine the diversity of innovation that currently exists in the field of UTI diagnostics. We then retained a patent lawyer and shared the progress we had made. And from this, we learned that we are too early in the innovation process to file a provisional patent, but this will be a priority of ours once we start prototyping and actually have IP to disclose. Another significant focus of our efforts was directed towards conducting market research to better understand the current state of the UTI diagnostics market. This provided us with an understanding of how the selection of a target market is interrelated with regulatory requirements and developing a go-to-market strategy. It also helped us to verify that there is a market need for the product we set out to create and that we have a unique selling proposition that differentiates us from competitors. Based on our market research findings and informal conversations with regulatory experts, We selected physicians in the United States who work at community clinics and cater to people with spinal cord injury as our initial target market. This did represent a significant pivot from our original vision of developing an at-home UTI test for people with spinal cord injury in Canada. However, this pivot was necessary when considering long-term commercial viability. As you can see here, the global urinalysis market has an estimated valuation of almost $4 billion, with the largest market capture held by the United States. Selecting the United States as the first jurisdiction to enter offers the most opportunity.
And all the work that we devoted to company development was done to support the eventual translation of our proof of principle study from bench to bedside. At the start of this project, our team conducted literature reviews to find studies on alternative UTI diagnostic methods. We found a number of studies that investigated the measurement of urinary immunoprotein concentration for UTI detection. From these studies, we identified 3 biomarkers that showed the most promise as novel indicators of UTI. There were, however, gaps in the literature that needed to be addressed to verify that immunoproteins could be used for UTI indication. First, we found that most of the studies on immunoproteins either recruited pediatric participants or older adults, but there wasn't a lot of research done with the general adult population. Similarly, we found that most of these studies did not take ABU into account. This is a particularly important gap that needed to be addressed. Our team decided to conduct a clinical study to address these gaps in the literature and determine the utility of immunoproteins in distinguishing UTI from ABU in the general adult population. In the fall of 2022, we were very fortunate to meet Dr. Dirk Lang and Victor Wong from the Stone Center at Vancouver General Hospital. They were very supportive of our project and offered to facilitate our pilot clinical study at their clinic through a service agreement and in-kind contributions. The purpose of this pilot clinical study was to determine whether the measurement of any of the 3 candidate urinary biomarkers could distinguish UTI from ABU, and whether these biomarkers could provide a more reliable indication of UTI compared to conventional urine dipsticks. Participant inclusion criteria consisted of those 18 years of age or older. Exclusion criteria consisted of those with severe immune deficiency, sepsis, genitourinary surgery in the last 3 months, and acute kidney injury. As a prospective observational study, there was no randomization or deviation from the participant's standard of care. As part of their regular appointment, participants were asked by their healthcare provider whether they experienced any of the UTI symptoms listed here. Relevant demographic information and medical history were acquired from the participants' EMR profiles. Collected urine samples were aliquoted, tested with conventional urine dipsticks, and then cultured to determine the concentration of bacteria. Based on symptoms and urinary bacterial concentration, participants were categorized into the UTI, ABU, and sterile urine cohorts. After the urine dipstick and urine culture tests, remaining urine samples were analyzed through ELISA to determine the concentration of urinary immunoproteins in the samples. The immunoprotein concentrations were normalized to creatinine to account for concentration differences. All de-identified data was entered into REDCap and analyzed using PRISM. So let's get into the results, the exciting part. Of the 68 participants enrolled, we processed samples from 8 participants with UTI, 8 with ABU, and 8 with sterile urine. For the purposes of biomarker analysis, only patients with above 100,000 colony-forming units per milliliter bacterial concentration were included in the UTI and ABU groups. This is the standard bacterial concentration threshold cited in clinical guidelines for the determination of UTI and ABU. So with this, 4 UTI samples and 5 ABU samples were included in the analysis. When positive leukocyte esterase and nitrite results were compared between UTI, ABU, and sterile urine groups, we found that positive leukocyte esterase and nitrite results were not predictive of UTI. These results align with the literature and further support that existing UTI tests are not reliable indicators of UTI. It was found that biomarker 1 levels were significantly higher in the UTI cohort compared to the sterile urine group. However, the concentration difference was not significant between the UTI cohort and the ABU cohort. For biomarker 2 and 3, significant differences in concentration could not be detected between the UTI and ABU cohorts, nor the UTI and sterile cohorts. As mentioned earlier, the notable result from the study so far is a significant difference in the urinary concentration of biomarker 1 between the UTI and sterile urine cohorts. This finding is consistent with results seen in other studies with pediatric populations. However, unlike previous studies, we were not able to demonstrate a significant difference in urinary concentration of biomarker 1 between the UTI and ABU groups. This could be explained by our limited sample size. Sample sizes reduce the power of the study, which limits the ability to detect smaller intergroup differences with statistical tools. We anticipate that more statistically significant results will be more easily detected when we analyze a larger sample size in the future. To be certain that biomarker 1 could serve as a novel UTI diagnostic marker, we would need evidence that this immunoprotein can distinguish between UTI and ABU. Another study limitation is regarding participant categorization. Given the challenges with UTI diagnosis, which we mentioned earlier, it is possible that some patients might have been placed in the wrong study category. While it is difficult to mitigate this error completely, participants in the study did not have impairments in communication or sensation, therefore we expect that they were able to describe their symptoms adequately. Another study limitation is that all participants were patients of a kidney stone clinic. Therefore, it is possible that patients' history of kidney stones affected immunoprotein levels in their urinary tract. Additionally, recruiting participants from one setting likely limited the diversity and the generalizability of this study. Future studies should recruit participants from other clinical settings to improve generalizability and to ensure greater representation of people who are vulnerable to complicated UTIs.
So where do we go from here? Thanks to this Capstone project, our startup has made great progress in its first year. With the start of the clinical study, we initiated the proof of principle stage of technology development. And in parallel, we analyzed the intellectual property landscape and the commercial market for gaps and opportunities, while also raising funds to finance these activities. In the coming months, we will continue the clinical study by recruiting more participants to increase the sample size, which we anticipate will help us acquire more robust results. We anticipate that the results of this clinical study will finally determine whether immunoproteins can serve as better indicators of UTI and whether we can move forward with developing this diagnostic method. We have achieved so much this year and have learned a lot, but we couldn't have done this alone. We want to thank our Capstone supervisors, Dr. Edyta Marcon and Dr. Richard Fody, as well as our program director, Dr. Joseph Fehrenbach, for their continued support and guidance throughout this entire process. A big thank you goes to our project advisory committee, Dr. Arrani Senthinatha, Dr. Ben Chu, and James Hektner, who met with us every month to help us overcome challenges and give us important perspectives to consider over the course of this project. Along with our academic supervisors and advisors, we are so thankful for the support and guidance we have received from our mentors at the Ideation Clinic, Health Innovation Hub, the Forge, and Praxis Spinal Cord Institute. And last but certainly not least, we want to thank Dr. Dirk Lang, Victor Wong, and the Stone Center Research Group for believing in us from day one and for helping us to make this project possible. In conclusion, this Capstone project has not only taught us valuable lessons in translational research and entrepreneurship, but it has also brought us one step closer to achieving our ultimate goal, which is to facilitate accurate UTI diagnosis for everyone. Thank you.